there, this is Sophie. I can't believe that I'm kind of at the end of the first phase of my journey in artificial intelligence. I won't say that I will not continue working on it, but actually my objective behind doing a focus on artificial intelligence and was really learning what I could do with it, learning how difficult it is to actually code artificial neural networks and discover what I would be able to do with it. So at the end, let's say the final result would be creating something that is as unique as possible and that is as useful as possible to humanity. So this was part of my brainstorming. I really wanted to leave it here just in case I want to actually build some of these ideas in the future. But let's, for the sake of simplicity, let's jump right into the actual project that I developed more or less during this month. Well, actually May and April, more or less. So these are kind of the milestones of the project. I'll guide you through the whole process, but first of all, I'll tell you what the problem is. So the problem is obesity. You know, there are millions of people with obesity in the world, and it actually leads to other diseases, right? So it makes sense to try to do something about it. And what's interesting is that throughout these couple of months, or yeah, not couple, but you know, throughout these past months, I've been really interested in microbes. So I didn't know how awesome they were, and I didn't know that they were almost everywhere. We have a skin microbiome, we have a gut microbiome, we have a vaginal microbiome, and men also have their microbiome. There's a microbiome for plants and a bunch of other microbiomes, and that's not the coolest thing. Actually, the coolest thing is that they actually do something useful. They can communicate with your brain through the gut microbiome axis. And the way they communicate is through chemistry, right? They produce certain substances that will tell your brain to do certain things. And this can have correlations with diseases or conditions like autism, depression, diabetes, cancer, and yes, obesity. So what I try to focus on is what food cravings these microbes induce and how that plays a role into obesity. So this is a little bit just of the scientific background. So scientists have indeed discovered that mice that are free from microbes behave differently to mice that do have microbes. And they've also discovered that the gut microbiome not only metabolizes what we eat, it also creates certain substances for us, even as essential amino acids, which is pretty impressive to me. What other things should we know? Yeah, they control somehow appetite hormones, either through immunological pathways or on their own, like directly, which is crazy again, thinking about obesity. So in terms of quantitative data, what this means is basically I wanted to get a sense of what specific substances we were talking about and what specific microbes are the most important and have the most correlation with obesity. I have like the list of bacteria here, and so sometimes, well, actually quite often, if not always, when we talk about the microbiome, we like to know the relative diversity. Yeah. So what this basically means is how what percentage of bacteria are present, what type of. So let's say we have bacteria A, so what percentage of it is present in the whole microbiome? That's kind of the summary. It's a little bit like generalized, but it's an easy way to understand what it refers to. And then here we have like also a list of different like correlations, like things, activities that you can do, like food you consume that relates to Shannon diversity, which is like how many different types of microorganisms you have in your gut microbiome. Because by the way, also diversity as a whole is really important in your gut microbiome. Like healthy microbiomes are often the most diverse ones. And then we have like which type of bacteria are like in a higher concentration somehow when you're underweight or overweight and so on. So yeah, I was just trying to get some background knowledge there. And now in terms of the idea itself, how this connects with artificial intelligence, I will tell you in a minute. Yeah, so my thought process for this project was, first of all, I wanted to do something simple, right? I wanted to take a look at the data and understand it myself, and then have artificial intelligence understand it somehow. So for this, let's say that the first phase of the project would be identifying or classifying actually healthy microbiomes from obese microbiomes. And for this, the very first thing that I did was looking for data sets, right? And I first looked for them on papers that had already worked on these correlations. It was difficult to find actual data that was useful there. So then I found a really good website that I'm going to leave in the description below. And from there, I had to download the data sets. I had to do some cleaning of the data. And then I was able to actually use it. But again, guiding you through my thought process, it was, first of all, getting data sets of obese microbiomes and labeling them as such. So it would be like supervised learning. Then I would do the same with healthy microbiomes, and I would join those two. And then I would train artificial intelligence based on the diversity numbers, the diversity, let's say, quantities of each type of bacteria, but also, the next phase would be training it to recognize full microbiomes, identify those like a healthy one from an obese one. So let me say that again. So one way in which AI would classify healthy and obese things, data, would be individual bacteria, right? So we have, bacteria A, which I know belongs to a healthy microbiome. Well, AI would have to learn if that concentration belongs to a healthy microbiome or not, and then it would do that with a whole set of bacteria, which contains bacteria A, B, C, D, E, F, G. And so that's what I did. I'm going to show you in a minute what the data looks like, but that's basically it. Then for next phases of the project, which I haven't actually started yet, it would be actually getting AI to understand, or maybe not understand, but at least know which bacteria are causing or could be causing this person to be obese. The third step would be knowing in which ways these bacteria are misaligned, meaning how much less concentration they would need in order to be healthy. You know, for example, if bacteria A, it has a concentration of 1.5, then AI should be able to identify, number one, that this bacteria belongs to an obese microbiome, an obese person. Then it would need to identify that this is the bacteria that is causing trouble to the person because it is in a higher concentration. And step number three, it would need to identify if that bacteria, how much less bacteria than the host would need to have in order to be healthy. So for example, I'll say like, yeah, you actually need to reduce this concentration by 0.1 or whatever it may be. And then based on that list, my kind of analysis was using reinforcement learning to teach AI to recommend a diet to people in order to, have like that correlation between what people will eat and how much that will help them reduce the concentration of bad bacteria or increase the concentration of good bacteria. And yeah, actually after that, I found out that colorectal cancer also has a correlation, like a very strong correlation with the gut microbiome. And that was pretty interesting, but then it was like starting another project. So I will now show you actually the code. So what I did was actually pretty lazy, I gotta acknowledge it. I pretty much just used pretty simple like models. A decision tree classifier is what I used at first. I imported some libraries to be able to do like these things, displaying the data and so on. So this is what the data set looks like. I had like the host, and you can see that the host is repeated because the host has different types of bacteria, right? So bacteria 0 would be, maybe it would be coli, but I needed to name it 0 as far as I could understand, because otherwise Python and, the computer wouldn't be able to understand it. Then relative abundance actually refers to the data that is useful for us. It is what is indicating whether this bacteria belongs to an obese or a healthy person. And finally, you know, preparing the data. I've mentioned this in previous videos. So it's kind of simple. We just need to know that 20% of the data will go for testing, and 80% of it will go for training. And yeah, like defining what the data set is. I actually of course have like a lot more data than just 3 rows or 4 rows, but it wasn't that much really because I wasn't able to obtain that much data. It was like 40 rows or something like that, which is like very poor for artificial intelligence to be able to make a good decision. But yeah, it was what I could do for an MVP maybe. And so yeah, it's part of splitting the data and then knowing if knowing if it worked. Like this step is actually unnecessary because then actually what I didn't explain is that the status refers to whether the person is obese or healthy. So 0 would be obese and then 1 would be healthy. So this step wasn't necessary anymore. In terms of creating the model, again, just syntax, making predictions, then checking, like comparing the actual values of the testing part versus the predictions that our AI made. And it actually turned out that AI got lucky this time and it got 100% accuracy. So I actually need to do more research into what this means in artificial intelligence, if it's like overfitted, or if, maybe I actually need to validate it with way more data. And this is part of like the other phase of the project. But then what I wanted to do was classify whole microbiomes, because by the way, this was only for individual bacteria, right? Like saying if E. coli is healthy or not in some way. And what I did for this was, again, importing certain things, preparing the data, but this time I used different models, and I actually discovered that the logistic regression model was the one that did the best with 75% of accuracy, which I mean is kind of bad, but again, considering that I didn't have that much data, well, I guess it more or less worked. And so I don't know if there's much else to explain, since these have been models that I've already used in the past. So if you want to learn more about these, then feel free to check out my other videos. And otherwise, I will end this video by saying that it was a pretty cool journey on artificial intelligence. It wasn't as difficult as I would have expected because I didn't get too deep into the math. I did understand the models, and I did understand, for example, when I worked with convolutional neural networks, it was actually pretty satisfying to actually know how the computer goes through all the process of filtering and so on. But yeah, I would say that definitely anyone can get involved in these sorts of things, and that intersecting this tool actually with biotechnology is going to be incredibly powerful. Also, I don't know, it's curious to me to get perspective on what's being done in the field, because sometimes, for example, with the colorectal cancer thing, I actually discovered that there were a ton of papers already trying to analyze this condition with artificial intelligence and trying to use this as a diagnostic tool for colorectal cancer, which is actually a really important type of cancer in the sense that it has like a high death rate, and a lot of people suffer from it. But yeah, I hope you learned something new. I definitely did, and I'll see you in the next one. Bye.